So I'm going to talk about vision, mostly. At the end, I'm going to try to squeeze in some auditory stuff. And when we look at an image like this, you see two cows, you see some grass, you see some mountains, and it's easy to forget that this is just a bunch of little tiny spots of different intensity and different color on a screen. And somehow we're magically converting that into this interpretation. So that's what we all want to figure out. How is it that we transform this into a sequence of representations that support recognition, that support all the things that we do visually, visual tasks, orientation, orienting oneself to the environment, et cetera, et cetera. So the question is, when the light enters into your eyes, and it goes into the retina, and then it goes down the optic nerve to the LGN, and then it goes to the back of your brain where your visual cortex begins processing it. Actually, it's already been processed quite a lot by the time it gets there. The question is, what is that sequence of transformations, and how does that give us vision? How is it that all those stages, those hierarchical stages that we sort of know about, how is it that they're leading to the process, the problem of vision? And I'm not going to talk about MRI in this talk, but a lot of the things I say will be kind of based on single-unit physiology. We know a lot about what stages of the visual system do from measurements like this, where people measure from individual neurons, record spike trains, and measure things like tuning curves. So in V1, we can characterize neurons according to their selectivity or tuning properties for various visual attributes. We know that individual cells are responsive to little tiny portions of the visual scene, a little tiny region that we usually refer to as a receptive field. So the question is going to be, how do we think about receptive fields and sequences of receptive fields as leading to perception? I'm going to go about that. So rather than just try to go ahead and answer that, I'm going to work essentially backwards, and you'll see what I mean, hopefully, in a minute. So if we go into V1, so this is a macaque, this is a lateral view of a macaque brain flipped the other way around. Sorry for the anatomists in the audience that are now confused. I flipped it around because I want the information flow to go left to right, and before I was going from the retina to V1, and now I'm going to go from V1 and beyond. Okay, so information comes into V1, and then where does it go? Well, we think for a long time now that it basically, roughly speaking, it's partitioned into two streams, a dorsal pathway and a ventral pathway. The dorsal pathway handles things like position and motion and things that support action, and the ventral pathway handles things like spatial form and recognition and memory. I'm going to be thinking about the ventral stream, as my title suggests, and the ventral stream has a bunch of areas in it, amongst others, these four. So starting with V1, there's V2, there's V4, and there's IT. So we know a whole lot about V1. What I mean by that is if you go in and you get a cell in V1, you can almost always drive the cell, get good responses out of it. You can almost always describe what it is selective for and what those selectivities are. You can measure tuning properties for spatial frequency, orientation, temporal frequency, et cetera, et cetera. And so we feel like we have a pretty good handle. We don't know all the details, but we have a pretty good handle on what those cells are representing. And similarly, we have a pretty decent idea of IT, and maybe that's because V1 is close to the stimulus and IT is close to maybe perception, or the things that we think vision is being used for, at least in the ventral stream. So IT, you can go in and you can find neurons that are selective for individual objects, maybe objects transformed or moved around in certain ways. And the stuff in between somehow remains a bit murky. And I think even the people in the audience that have worked on this very hard for many years will admit that we really don't have the level of understanding of these intermediate areas that we have, certainly not that we have in V1, and probably not even, we can't even say as clearly what they're doing as we can do for IT. So the question is, what's going on in the middle? And that's what I'm going to try to think about. These are just some examples of papers where people have been able to measure selectivity properties of cells in these areas and have made some progress. So they'll find maybe a subset of cells that they can drive reasonably well and that have properties that can be related to the stimulus. They can talk about tuning for orientation or maybe combinations of orientation. But still, there remain lots of cells that you can't drive and lots of cells that you can't really explain. So I think we're still not at a point where we can say, here's what V2 does or here's what V4 does. So I'm going to go about this in a kind of funny way. It's not the normal way you would do this, where you think, okay, I want to transform things from the retina to V1, and then I want to end up with something that's selective for objects. How am I going to build that? Well, I'm going to build a sequence of transformations that are deliberately allowing representation of structures by combining substructures. I'm not going to do it that way. I'm going to basically put on blinders. I'm going to forget that I know anything about objects or recognition or anything like that. And I'm going to say, what are the basic things that we know about the system? I'm going to use some simple facts, that it comes in a hierarchical cascade, that there's variation in receptive field sizes, and that I'm going to assume that these areas are performing some sort of canonical transformation, like the same kind of transformation is being done in each successive area. And by putting those things together with one other thing, which is kind of under the rug here, which is some thinking that we've done in my group over many years about representation of texture, we're going to be able to build a model for ventral stream, and then we'll be able to show that that can make very strong and clean predictions about deficits in the periphery. Okay, so that's basically the plan for the talk. So let me go through these constraints that we're going to use to build this model, and then we'll ask, we'll put the thing together, and then we'll say, okay, what does this compute? What can we do with this? Okay? So first constraint, color is really strange on this projector, at least on this slide. Okay, so this is a diagram that represents the visual hierarchy. The size of the boxes show you roughly how much cortical area is associated with each of these, how much cortical volume, let's say, is associated with each of these visual areas. And the black lines are indicating the connectivity. Some of these are still debated, and there's a lot of question about exactly what these connections are, but let's just take it as it is, okay? So what we see is V1 is here. V2 gets a really large input from V1. V1 is also a very large area. V4 gets some input from V2, but also from V1. And so when we think about what's going on in the ventral stream, let's say the mid-ventral stream, V2 and V4, it's going to be hard to say whether we want to talk about V2 or V4, since they're both taking some input from V1. And V4, even the input that's coming through V2 is kind of effectively still based on V1. So it's going to be hard to think about which area we want to talk about. So I'm going to kind of remain agnostic. I'm going to build a model that's generic for a ventral stream that calculates things based on V1 responses, and then we'll ask, what does the model correspond to at the end? Okay? So that's the first point. Things are hierarchical. They go in a cascade. Here's the second point. These different areas, one thing that's been known for a long time, actually, is that the receptive fields in different areas are different sizes. And so you can see in this plot, this is a plot of some data from macaque. And these are receptive field sizes as a function of eccentricity, that is distance from the center of gaze. And what you see is that for each of these areas, V1, V2, and V4, things go up roughly linearly. The sizes grow. This is linear size, not area. Okay? So the diameter of the receptive fields is growing roughly linearly with eccentricity. And you can also see that at any given eccentricity, V2 is about twice the size of V1, and receptive fields in V4 are about twice the size of V2. Okay, so we have this size distinction between the areas, and we can use this essentially as a signature or a description of the different areas. So that's going to be our hook at the end to get back and figure out what part of the brain we're talking about. We're going to try to ask what size of the receptive fields, the effective receptive fields.
How were the receptive field size measured?
Right, so there's a lot of questions about that. What I'm going to show you at the end is compiled from data from a very large set of studies, 9 different studies. So we've taken monkey data from 9 different labs. They're measured in different ways. We did the best we could to try to reconcile those differences. This particular study I actually don't remember, but I should have said at the beginning that a lot of the work that I'm going to tell you about, not all of it, but a lot of it is done with Jeremy Freeman, who's here in the audience. He's my junior colleague that came along to the meeting, and he can give you the details about, probably, do you remember? The details about exactly how these were measured.
I think just with gratings, simple area summation with gratings probably optimized for each cell, so pick the optimal orientation and frequency, and then do an area summation curve.
So there are questions about that. But I think this fact seems pretty robust across these studies. And it's somewhat supported by MRI data in humans. That's a little bit complicated because it's hard to measure receptive field sizes with MRI. But people have tried, and they've measured essentially effective receptive field sizes. And they come out, I would say, consistent with this. Okay? All right, so this is our second fact that we're going to try to exploit, the fact that receptive field sizes get bigger through the hierarchy and also with eccentricity. Okay? So a cartoon of that might look something like this. In V1, we have a pile of this. This is just a cartoon, but just to illustrate the idea. You have a pile of receptive fields radiating outward. They get bigger and bigger as they go away from the fovea, which is in the center of the diagram. Here's V2. The receptive fields also do the same thing, but they're bigger. So at any eccentricity, they're bigger than they are here. And the same thing in V4. And by the time you get to IT, the receptive fields are absolutely huge. A lot of them cover the fovea. Many of them are nearly the entire visual field. Okay. So, and by the way, so this scaling of size with eccentricity is not something that starts in V1. In fact, it might be that you can explain the whole thing as originating back in the retina. So this is a little diagram that I made after, sort of based on data from these two papers, that shows the size of midget ganglion cell receptive fields in macaque. The sizes are actually multiplied by a factor of 10 here so that you can see them, because otherwise it would be so small that you wouldn't really see the effect. And kind of nicely enough, Stuart Anstis many, many years ago pointed out that receptive field sizes in retina were about the right size to explain visual acuity. So what's shown here is a Snellen acuity chart arranged so that you're supposed to fixate on the red point in the middle, and each of the letters is at a size which makes it equally readable if you're fixated here. Now, these are 10 times the threshold for typical acuity, 20/20 vision. But you can see, since I multiplied both of these by 10, the sizes of the strokes are about the size of the receptive field. So it kind of looks, at least roughly, like it works. Like you can explain the fact that these letters are each at threshold based on the sizes of those receptive fields. Okay. So this thing starts in the retina. And it might be that, in terms of the construction of the visual system, it just travels its way through the hierarchy with each region basically pooling over chunks of the stuff that are coming from the previous region, but pooling over larger and larger regions. And the larger and larger regions are coming just because basically how do I say this? If V1 grabs a fixed number of these retinal ganglion cells, when it's here, when you're in here near the fovea, and it does the same thing here, grabs the same number of ganglion cells, then you will get the effect that I described. That is, the V1 cell will always be a fixed ratio larger than these receptive fields in the ganglion cells, and then V2 could do the same thing again. So you get a constant ratio between areas, and everybody would scale linearly starting from the retina up. Okay.
Just a quick curiosity.
Yes.
Size doubles at which eccentricity?
Size doubles at which eccentricity?
5 degrees, or I should know.
Yeah, I'm not remembering the numbers. Let's see if I wrote it in my notes here. So these guys, so for V1, for V2, the scaling is about 0.5. For V1, it's about 0.25. And these are about 30 times smaller than V1. So the scale factor is going to be about 0.01, let's say. Okay? So what I mean by that is the size of the receptive field is 0.01 times the eccentricity. So from that, we can work it out. But you won't. Your question was, again, where do you get half?
Yes. At which point, if I resize it in the retina, circle in the fovea,
Yes.
at what eccentricity I get?
Double the resolution.
Double the size of the...
Yeah, so there's, it's hard to answer because, and I left a hole here, because what happens at the fovea, of course, is they don't go to zero. So you have to decide how you're going to handle the fovea. In the plot that I showed you before, we actually used what we call a hinged line to fit the data. So it's a straight line to the origin, but right near the origin, it goes flat. And that's certainly true. If you think about cone packing in the retina, that has to be true. The cones have a physical size. In the fovea, they're actually packed right up next to each other. Oh, I changed my changed my slide. That was pretty good. They're packed right up next to each other in a hexagonal lattice, and when you go out, when you go out away from the fovea, they spread out. So they can't go down to 0. It obviously something has to give near the origin. Okay. Okay, so, and if you put the retina on this slide, it's way, way down there. I said a minute ago, it's a factor of 30. The ratio between the slope of this guy and the slope in V1 is a factor of 30. Okay, so back to this picture. So how are we going to use this? Okay, so the intuition that you have when you think about that is that, well, these receptive fields are essentially pooling, or just think something really simple and stupid. They're averaging information over little regions of the visual field. And in the retina, that's not that far from the truth. They're basically averaging over a clump of cones. And their center surround. It's not really an average, but it's close enough to an average. You might even think of it as just adding up the cones. Okay. What about, so if that's all it is, then why does the visual system do this? And what would be the point of averaging over bigger and bigger regions? Well, and, the obvious answer is that it's not just averaging intensities. And as you go deeper and deeper into the system, what you're averaging are things that are more complicated, because in the previous stage, they've gone through some nonlinearities, and so what you're averaging are things that are more complicated. So the question is, what are we averaging? So to finish the model, we're going to have to think about what's inside of each of these receptive fields. And there, I'm going to do really the simplest and most basic thing you can imagine, and it goes really back to Hubel and Wiesel. So this is a diagram from one of the original Hubel and Wiesel papers, 1962, where they tried to say, and they weren't being computational or mathematical about this, they were just saying loosely, look, the way you build a V1 simple cell is, here's your V1 simple cell, it basically just adds up the responses, or they didn't say adds up, combines the responses, that is, receives afferent input from a set of LGN cells. And the LGN cells are cells that are selected to have their receptive fields aligned along some straight line. Okay, so here's the receptive fields of the LGN cells. They're lined up, and you just combine those, and that gives you your V1 simple cell. Okay, and a more computational version of that might look something like this, and a lot of models, including all of the sort of classic neural network stuff, is based on this kind of a model. So the idea is you take these LGN cells, an array of them, you weight them with linear weights, so the gray scale here is supposed to be a weight. Multiply each of these guys by the associated weight, add them up. That's a dot product if you want to be mathy about it. Out of that comes a number. Now I have a linear combination of these guys, and then just run it through a rectifying nonlinearity, and that, if you want to be biophysical about it, that rectifying nonlinearity is just something that's converting membrane voltage into firing rate. Okay? So, and out of this comes spikes. You have to do some sort of spike generation. This is really simple, but it actually captures a lot about the behavior of these cells. And so the other thing that Hubel and Wiesel pointed out is they said, oh, well, there are these other cells that we're going to call complex cells, and in the same paper. And they pointed out that the complex cells had this property that, well, the simple cells, you can find positive and negative regions of positive excitation and regions that suppress the cell. And in the complex cells, that didn't quite work. It looked like what the cells cared about was having something of the right orientation, but it could be anywhere. And it didn't matter whether you put in stuff that was positive or negative, that is, light below or above the mean luminance. Both of those would drive the cells well. And so they postulated that you could construct such a thing by taking a bunch of simple cells, all with the same orientation, and combining them. And so they used, again, this kind of a diagram. This is their hand-drawn diagram of this. You basically take a bunch of simple cells and combine them, and that gives you a complex cell. And so computationally, we can do something like this. We can take a bunch of these linear weights, and we can take shifted versions of them. Here, they're shifted horizontally. So here's one, here's another, here's another. And then we can rectify all of those. Those are each of those is a simple cell, and we can add them up, and that's a complex cell. And this basically is you can find this a classic paper that describes this construction, not exactly this construction, but something quite similar, is the Adelson-Bergen paper in 1985 about the energy model. And lots of other models have done similar kinds of things to this. So we're going to take this as a generic calculation, add up a bunch of things with different linear weighting functions, and then rectify them, and then add them together at the end. Okay? And we're going to ask what can we compute with that. But I'm going to make this assumption that, so this is V1. I'm going to make the assumption that V2 is just going to do the same thing. But instead of doing it on LGN afferents, it's going to do it on V1 afferents, obviously. Okay, sorry, and I shouldn't have said V2. I'm supposed to be agnostic about that. I should have said someplace in the middle of the ventral stream, we're going to do the same thing. We're going to add up. I've ruined my punchline. So we're going to combine responses coming from V1, afferents coming from V1, and we're going to take multiple linear combinations of those, rectify them, and add them up. Okay. So if these are the ventral stream receptive fields, the idea is that inside of each of these, there's an array of V1 cells, and we're going to add them up. That would give us, if we did it this way, that would give us a ventral stream simple cell, and if we did it the other way, we would get a ventral stream complex cell. So this idea, this is not a new idea. This is, you can see this in papers going back on the computational side, work like Fukushima's work or Tommy's work, talking about cascades of simple and complex cells in a hierarchy. And there's also a lot of neuroscientists that have mentioned these kinds of ideas. So the question is going to be, if you do this, what does it compute? What do you do? What are you getting out of this? If you build something like that, why is it useful, and what is it representing? And it's not completely obvious, I think, because I should point out, we're going to do this. These linear combinations are not just going to be over a bank of V1 cells all at the same orientation, but we'll also take combinations across different orientations and across different sizes, different scales. And so the question is, if you take a collection of those things, what is it that you've represented, and how do you think about that? Oops. There we go. And the other thing is, well, once you put this model together, what does it represent, and how do you test it? How do you verify that you actually have something that's relevant, that is meaningful in terms of human vision? Okay. So now I'm going to go through a bunch of stuff about how you test this and how to think about what it represents. And to do that, we're going to have to use this concept of a metamer, and that was in my title. But I realized that there might be, how many people in the audience know what a metamer is? So about how many people don't know what a metamer is? Don't be embarrassed. I wasn't born knowing what a metamer is. Okay, good. So I've decided to tell you a little bit about metamers so you know what they are, so I don't go through the rest of the talk with you sitting thinking, this makes no sense. Okay. So this is, well, I'm asking you if you know what a metamer is after I put the definition up. That was good. So here's the definition. Two stimuli that are physically different but appear the same to a human observer. We call those metamers. And the term comes, the strongest use of this term and the oldest use of this term, I believe, comes from trichromacy. So for those of you that study vision, I hope you've heard about the history of the story of trichromacy. And if you haven't, I would urge you to go and read about it. It's in my view, it is absolutely the most beautiful story that we have as a community, the vision science community. It is a story that unfolds over hundreds of years, and it's a model, in my mind, for how perception and computation and the underlying implementation, the physiology, can come together. Well, so let me just, I'll tell you quickly what that story is, like in 3 very quick slides, because it's so amazing that you have to know about this, okay? This is not my work. 1665, I am getting gray, but I'm not quite that old. So Isaac Newton figures out that light is made up, is actually assembled from bits of colored stuff, right? So he realizes that you can take light and you can split it into spectral components. And he does that. This is a diagram of what he did. He basically opens a hole in the wall, lets the sunlight come in, runs it through a prism, and notices that he gets these colored stripes. He also goes through a whole set of experiments pointing out, learning that you can't further divide it. So if you take a little chunk of this and run it through another hole and try to do it again, it doesn't split again. That's it. He also recognizes that he can reassemble it. So he can take all those colors, pack them back together, and get back something white or yellowish. Okay? So critical, 1600s. Okay? So then, in the 1800s, a bunch of scientists, they're mostly physicists, but they're kind of psychophysicists because they're thinking about perception, realized that actually humans can't see all that. And in fact, if you give humans, if you put them in an experiment where on one side they're looking at an arbitrary test light with an arbitrary wavelength spectrum, and on the other side you use 3 fixed lights that have fixed wavelength spectra, you can adjust the strength of those 3 fixed lights to produce a light that looks identical to the one on the other side. And for normal human observers, they can always do this task. They can repeat it with identical settings across multiple days, and all humans pick the same settings. I say all. There's always caveats. There are people that are colorblind that give different answers, and there's always noise. But basically, the simple version of it is you only need 3 colors. And you all know this because we have devices like this that have 3 colors, right? And we have TV screens and we have printers and things that can get away with 3 colors. So why is that? It's because your visual system at the very front end is discarding a huge amount of information. It's taking the entire wavelength spectrum and smashing it down into 3 numbers. We know that now. In the middle of the 1800s, they didn't know that. But they noticed, by making a lot of careful measurements, that humans really could only see. You only needed 3 things to reproduce any color. And this took decades to work this out. But they worked out not only the experimental observations, but they worked out a full set of, a theoretical set of descriptions that captured this. And basically, Grassmann in particular invented chunks of linear algebra to explain the data that he was getting out of the psychophysics. So this was a day when you could do math and perception and physics all at once. Okay. So that's middle of the 1800s. And the amazing thing about it is they totally nailed it. And more than 100 years later, so this is an example of a set of wavelength spectra, and this is a matching. This is an example from a color matching experiment where here are the 3 primaries, the red, the greenish, and the bluish, and the black thing is a test light on the other side. And to you, those 2 things, the right combination of these 3 guys, adding these 3 guys together, would look exactly like this, even though obviously those 3 curves are not going to add up to look like the black curve, right? Just to make the point. So physically, they're very different, but perceptually, they look identical. Okay? So that's a metamer. And just to finish my punchline, more than 100 years later, Dennis Baylor, along with colleagues, figures out a way to suck up a cone in, this is, I think, from a macaque monkey, in a glass micropipette, shine light through it, and measure its spectral absorption. And he finds that the absorption properties, there are 3 kinds of cones, and the absorption properties of those 3 kinds of cones are perfectly covering the same space that was worked out 100 years earlier. So this is an amazing triumph, right? We have perception and computational modeling in the 1800s making a near perfect prediction of what's going to happen 100 years later when we can finally measure the actual mechanism. Okay. I'm done with my advertisement for Isaac Newton. If you haven't heard about this story, you have to read about this because it's a bedrock of the field, and it's a fantastic and beautiful example of how we can make things happen in vision science by bringing these different kinds of measurement together and different kinds of tools. Okay. Those are the measurements that they made of absorption. Okay, now I mentioned that there's another example of metamers, which is not usually called metamers, but I think it's really the same thing, and that's texture. And that one really gets credited to Bailey-Ulej. In 1962, he postulated that 2 textures would look the same to a human if they had matching pixel statistics of a particular order. And he wasn't sure what the order should be, but he made a very general theoretical statement. And then he set about trying to test this. And the amazing thing about this is, so first he had a very explicit concept that you could capture perception with a small number of measurements. Okay? So just like the cones in color, you make 3 measurements and that captures perception. He's doing the same thing. He's saying you measure a small number of statistics and that captures everything you can see. Anything else, you can't see. Okay? And that's why 2 textures that match in terms of those statistics will look the same. So he's coming out, he's using the same concept. He didn't call it a metamer, but it's the same idea. And he also comes up with a conceptual framework for testing this idea, which involves generating images that have identical statistics up to some order, and then showing them to himself, basically, and asking, do they look the same or do they not? Can I tell the difference between these things? And so he uses that to falsify his own theory for n equals 2 and n equals 3, and then he gives up. And he gives up partly because he thinks, well, it probably doesn't work, and partly because he can't go higher than n equals 3 because he's generating these things by hand. So he doesn't have a computer like we have now that can spit these things out and generate them automatically, which is what I'm about to show you. He's trying to do it by hand, and after 3, he says, this is too hard, my brain hurts, I give up, throws the theory out, and goes on to a very different theory of texture, which, frankly, in my view, is much less successful, although it's certainly very well known, and that's the theory of textons. Okay. I knew I would get a remark from the audience. Go ahead. Okay. All right, so now back to where I was. Okay, so now I hope you understand roughly what a metamer is. I hope you have some sense of how it's been used in the history of vision science, at least for these 2 examples. And now I want to get back to the ventral stream, and I'm going to make metamers, and I'm going to use them to test the model. Okay, so here's the idea. Okay, so we're going to take an image. We're going to put together this model of ventral stream receptive fields, and we're going to measure a set of numbers corresponding to all those mid-ventral complex cell responses. So we're going to collect an array of numbers. And we're going to do that not only for different combinations of the V1 cells, but we're also going to do it over each of these receptive fields that are of some size appropriate for the ventral stream. Then we're going to come over here, and we're going to synthesize a new image. We're going to start with an image of white noise, and then we're going to push on it until it produces the same model responses. Okay? We're going to generate a random image. You can think of it as there's a space of images that produce the same model responses. We're going to pluck one randomly. And we're going to do that by starting at some random position and pushing ourselves onto that collection until we have landed on one. And then we're going to say, OK, here's an example of something that produces the same responses. And we're going to synthesize images that way. And each time we do this, we're going to start with a different white noise sample, and we'll get a different synthetic image. Okay? So we can generate a whole bunch of these. And if the theory is right, so here's the experimental logic. So here's your original image. You measure your ventral model responses. Here's a random seed image, white noise. We're going to push on that white noise until it matches those ventral model responses. And then we'll show the 2 things to a human. And if the theory is right, or if the model is right, by the way, I'm sticking in one additional assumption. So we have this model for the ventral stream. I'm going to assume that if it's right, that a human looking at images can't see anything other than what's coming through that model. They don't have any other source of information. There's no other pathway that they can look at. That's it. They're not able to consciously access or use information that is not coming through that model. Okay? So if that's true and the model's correct, then 2 images that have identical model responses should be indistinguishable. They should be metamers. Okay? All right. So we're going to test that, and I'll jump to the punchline and I'll show you some metamers. So here's an image of a crowd. Jeremy took this in Washington Square Park. Typical scene there. So here's a fixation point in the center, and this only works. So remember, this whole thing is built up with receptive fields that get larger the further they are from the fovea. So this is only going to work if you put your center of gaze on that point, so your fovea is in the right place, and your receptive fields are matched up to those of the model. And if the model is right, you shouldn't be able to tell the difference between the images I'm about to show you. So what I'm going to do is I'm going to put up images that are going to flash back and forth. And if you keep your gaze fixed right on the red point, you should not be able to tell the difference between the 2 images that are being flashed up. But if you look away, so now you're breaking the model because you're moving your eyes somewhere else, this red ring is about the size of one of the ventral stream receptive fields. And you can see that the content inside of there in the original image is a face, several faces, one guy with his hand over his head like this. And in the synthetic image is a scrambled mess. Okay? But you can't tell the difference if you look at the fixation, if you keep your gaze centered over here. Okay?
Eero, where does the face come into this?
Where does
Phase.
phase?
Phase.
The full answer to the question is a little complicated because phase and energy do not separate as cleanly as they do with a global Fourier transform. We're talking about local filtering operations. And so phase is partially represented in here by the complex cell responses. But it is definitely mixed up. And that's why boundaries and contours get moved around a lot within these metamers. When you generate these metamers, stuff gets scrambled and moved around, and yet you see content that looks like contours and edges, and things have the right color and basic intensities, and that's why you can't tell. So the phase loss is what it is.
But there is no phase loss in the fovea, right?
Correct, because the receptive fields in the fovea are very tiny, and so effectively they get down to the point where they're nearly the size of the pixels in this image, and at that point you're reproducing the original image. So in fact, the center of these images is identical, physically identical. The pixels are the same. The stuff out in the periphery, very different, as you can see by looking at the content inside the red ring. Yeah?
Sorry, quick question. Do you think that's a tiling effect? Are you actually shifting your receptive field at all, or are you having a fixed tiling?
So the theory, the model is that they're fixed. I should point out, those toy cartoon pictures I showed you of receptive fields were little circles with gaps in between them. The real model is not built that way. These tile uniformly and smoothly, and they overlap. Okay, so they're not independent. They're overlapping, and they're smoothly covering the whole space. Okay, and surely that's what is happening in here too. You don't have big holes except at the blind spot. That raises another issue, but anyway, let's keep going. Okay, everybody had long enough to look at this. It's driving you crazy now, or really, really bothersome the fact that you can't see anything in your periphery. Yes.
Can't you make a metamer in the fovea?
I think you probably could if you did things at high enough resolution, but I can't prove that to you.
Yeah, it may be impossible.
So certainly there's information loss in the fovea. The color stuff that I mentioned, the trichromacy thing, is happening in the fovea too. So there is information loss. So there are in that general sense, there are metamers in the fovea. Can we make a metamer like these metamers that loses local sort of feature structural organization? Maybe not. I'm not sure. There's a murky, so you've uncovered one of the caveats, which is that in this entire modeling endeavor, I'm waving my hands. I'm waving my hands a lot because it's my Italian origins. But sorry, Tommy. But in the fovea, we don't know exactly what's happening in the fovea. So something has to break there. You can't go down to zero. Somehow you have to roll it off and reach a constant resolution. And we don't actually know how that propagates through the hierarchy. And so we're fudging a little bit about what we do there. And I think it's a really interesting question. But I don't think that we have precise enough measurements to answer it right now. And either on the physiology side or on the perceptual side. So I think it's an important topic, but I can't really give you much of an answer. Okay.
Is there any reference? We didn't see the difference between the metamers, but do you have another transformation to show that we do see the differences if it's not according to your...
Yes, well, in a sense, the experiment is going to do that. So I think I'll answer your question right now, okay? All right, so we're going to... We didn't want to just... This is not based on just casual viewing. We did an experiment, a couple of experiments. I should say Jeremy did an experiment. Here's the experiment, the basic experiment. We're going to take the model and we're going to adjust. We're going to pretend that we don't know where this is happening. And we're going to adjust the receptive field sizes in the model. In particular, not going to adjust each individual receptive field size. We're going to adjust the scaling. So the slope of that line that describes how things vary with eccentricity, we're going to adjust that. Okay? And but we're going to do the same calculations within the receptive fields. We're just going to change the size of the pooling regions in the ventral stage. And the question is going to be, if we make the line too shallow, so the receptive fields are very small, then within each of those regions, we're not actually averaging over much. And when you build a metamer, then the values will end up being very, very closely matched at the level of V1. And so the images will end up being quite similar. If you let the receptive fields get really big, then a lot of scrambling will happen because there are many images that produce the same average values within those receptive fields. And then you're going to get gross distortions. So as you change this axis, changing the size of the receptive fields in the model, you go from limited distortion in the periphery that's happening, for example, in this image, up to really gross distortion in the periphery that's happening in this image. And what we can do is examine the human capability to distinguish the two images as a function of that model parameter. Okay? And what you can predict is, well, over here on the left, humans won't be able to tell the difference between the two images because this looks pretty close to the original, and their performance at distinguishing will be around 50%. And over on the right, as the distortions get more and more wild, you'll be able to tell the difference much more readily, and then their performance will go toward 100%. And that's exactly what happens. So here's an example from a single subject, and you can see that the performance is going from 50 up toward 100%. The shaded area is error bars, and we can actually predict this shape by taking the same model. This is a little bit complicated to get the logic, but we use this model to generate the images, but now we're going to use the same model as a model for an observer and ask, if I were making those measurements, what would the observer be able to distinguish? And we can actually predict that the shape of this curve should be of this form. So this is a cumulative Gaussian, and it's a function of 1 minus the scaling factor for the human divided by the scaling factor for the model that was used to generate the stimuli. The point of all of that is, first of all, this thing is going to have a couple of parameters, but the most important one is this one, S human. What we're going to do is we're going to fit the data. We're going to fit, so S model is just this axis. We're going to fit this to the human data, and by doing that, we're going to have to select an S human that fits the data, and that's going to tell us the size of the receptive fields in the human. Okay? So it's complicated to get the, I didn't hear that. Okay, so we're going to use this to figure out how big the receptive fields are that the human is using in this task. Okay, so the bottom line is we do that. Here's 4 observers. There's an average of the observers. First, they come out consistent. Second, and they come out consistently around 0.5. And nicely, so this is what I was mentioning earlier about the macaque data. Here's a large set of studies of macaque physiology that Jeremy carefully and painstakingly went through and pooled and kind of unified in terms of the techniques that were used to actually measure the receptive field sizes. And these color bars indicate the receptive field sizes, or really the scaling of the receptive field sizes for the different areas. And the height of each bar is basically telling you the error bars from a bootstrap analysis of all these data sets. And what you can see is our observers are squarely and clearly in the V2 range. Okay? So we conclude from all of this that this model can produce metamers. It really works. And it works when you set the receptive field sizes to the size of receptive fields in V2. So from all of that, we can arrive at, well, we think we have a description of at least part of what's going on in V2. I don't want to claim that this is a full description of V2, and it's kind of a loose model. There's a lot of wiggle room here for refinement, but I think it's a really nice step forward in terms of understanding what might be going on there. And just to mention, so a lot of you are interested in MRI, and you know a lot about things like attention and cognitive things that I know nothing about. We have done a control for cognition. So we did a task where the observers had a cue that directed their attention when they were doing this task to the part of the image where the change was biggest. And it turns out that improved their performance, but it did not significantly change the estimate of the size of the receptive fields. And we think that that's really important because we want to think of this as an architectural limitation of the system. The same way in color vision, the reason you can't see colors other than those, what's projected under your 3 cones is because the information is completely lost by the architecture, and no matter how hard you concentrate or attend or think about the stimulus, you can't recover that information. It's gone. And so we want to think about this the same way. The idea is that V2 is losing information. It's discarding information. And that is built into the architecture of these receptive fields and their size dependence on eccentricity. Once the information is gone, you cannot recover it no matter how hard you try. Attention can improve your performance, so your percent correct goes up. The curve kind of scales up, but it doesn't shift left or right. You don't get a change in the receptive field size, the estimated receptive field size, by giving people an attentional cue. Similarly, giving them a longer presentation, just letting them look at the stimulus longer in the experiment, also improved their performance but didn't change the receptive field sizes. So we feel reasonably confident that this is an indication that we're really capturing something in V2. Okay, so let me say a little bit about perception and then I'll end. So some of you may know about this thing called crowding, perceptual observation that's been made starting in the 70s. And the idea is that when I fixate here, I can see this letter, and if I put 3 letters together and I fixate here, I can see all 3 of these letters. But if I move them out here, it's actually really hard to read that H. In fact, generally you can't. People refer to this as crowding, and it was first noticed with letters, and a lot of the studies are done with letters. Later on, it was extended to other kinds of stimuli. But the basic observation is that when you mix together stuff in the periphery, it interferes in some way that's hard to figure out, but it prevents you from recognizing or identifying things. And crowding has rules that it basically, the regions over which crowding happens grow with eccentricity. So there's a nice parallel here in this perceptual literature, and recent perceptual literature, and especially this last paper by Ruth Rosenholz and her group, postulates that what's really going on is that you're computing some sort of statistical texture-like representation out here, like an average of stuff. And once you've averaged, you can't pull things apart again. And so the model that I just described to you basically takes that a step further by building a full global description of the visual field and actually generating these metamers. So we can show examples of crowding. So these are single letters, and if we synthesize an image to produce the same responses in the model, the single letters actually come out pretty intact. They're pretty readable. If we bunch up a collection of letters like this, they interfere with each other in a dramatic way. And you basically can't read any of these. So we can simulate crowding using this model, and we get the eccentricity behavior completely. So when you move these closer, they don't interfere as much because the receptive fields are smaller and they're not pooling over multiple letters. Okay? So all of this comes out of this model that's kind of built on the physiology. And so we think that this has a lot to do with reading, and I'm not the first person to say this. There are people that have proposed that things like dyslexia are related to crowding. But we can now, the thing that the model allows us to do is we can now simulate this in a global way. So I can take a whole page of text, here's a little chunk, and I can build a metamer for that, and I can show you kind of roughly what I don't really want to say this is what you see, but this is another image that's indistinguishable from the actual page of text is the right way to say it. It's a metamer for the true page of text. And what you learn from that is, well, you can't read anything that's more than about 5 letters away from this fixation point. Okay? And the way you read, of course, is by hopping your eyes, fixation to fixation. If I jump to approximate the next location of a fixation when you're reading this text, it would be about here, and you can see that it works out just about right, that these words, these letters become unreadable right about the point where these become readable. So we think we can explain something about reading and about fixational hopping while you're reading, and out of that comes a whole lot of nice ideas, like maybe you can adjust the spacing or the details of fonts in a way that causes less crowding. Now that you have a model that can measure how much of this effect is going on, you can adjust the properties of the stimulus to actually reduce the effect. And it might be possible, I don't want to, this is a speculation, but it might be possible and interesting to explore the effects, the readability of different fonts, for example, as a function of how much crowding occurs. Here's another example. This is a soldier in Afghanistan, so camouflage. If you look right at him, so these little black marks tell you where the fixation is for the model. If you look right here at his head, it's obvious that he's there. And if you look away over here, he's kind of blended into the background. So the point is that the camouflage works because most of the time you're not looking right at him, and when you're not looking right at him, you can't see him. So maybe we can design better camouflage by arranging that it basically blend into the background using this kind of model as a measure of how well it's blending. Anyway, these are just some loose ideas about what one might do. I'll just mention this, because I don't have time to play you the examples. If you want to hear them during the question time, I'm happy to do it. We've done a similar kind of thing for the auditory system. So with postdoc Josh McDermott, we've taken sounds, decomposed them in a model that's got a cochlear filter bank, followed by some nonlinearities, followed by some modulation filtering, and then a bunch of statistics. All these little red circles are just statistical measures, little averages of stuff across bands and within bands. We take that, we take a sound, we generate a metamer, and then we did a whole bunch of psychophysics to determine whether people could tell the difference between these things as a function of which statistics we included or didn't include. And we've been able to build this into a model for sort of early stage auditory system representation of these kinds of sounds. So this is just to point out that I think this is a generic approach, and I think it works across different modalities because I think the same kind of computations are going on in different cortical areas. That idea of a canonical computation happening in cortex is an old one, and I think we now are at a point in the evolution of the field where we have the computing capabilities to actually test it and push on it. So that's what we're trying to do. Okay, so let's see. What else do I need to tell you? Just put up my concluding slide because I'm not going to play the sounds. And I want to, so the work that I told you about, the original model of texture that a lot of this stuff was based on was done with Javier Portilla about 10 years ago, who's a postdoc in my lab. A lot of the work that I told you about with ventral stream metamers was done with Jeremy Freeman, who's here, and he's got a poster. We're doing now some physiology in V2 to try to measure individual cell responses to these kind of textures to understand in more detail exactly what is being represented by individual neurons. And he's going to have some of that on his poster if some of you would like to see some of that. I imagine you'll find it interesting. Okay, the auditory modeling with Josh McDermott and the V2 physiology I mentioned just a minute ago. And that's also with another grad student, Corey Ziemba, who's not here. And of course, all the physiology is not done in my lab. I'm a theorist. That's done in Tony Movshin's lab. Okay. And I guess as a segue to the next part of the coming speakers. So, I started out talking about, I put out these two cows. I said, how do you know those are two cows? All you've got are little colored spots landing on the back of your eye, and you somehow assemble that into, you do recognition. So these kinds of generic calculations I just told you about, I just went through this story that says if you pile these up and then you ask what is it representing, it ends up representing these scrambled texture-like things. So now, if you go back to the beginning and you say, well, wait a second, what does that have to do with object recognition? Can you use these sort of texture-like or statistical representations out in the periphery where you've pooled or averaged over large chunks of stuff? Can you use that to actually represent objects? I don't know the answer to that, but I think it's a really interesting thing to think about. You can imagine that, objects are represented in a separate stream, and this is a stream that's more textury, or you can say, maybe this is a substrate for object recognition, and we just don't understand how to use it. And there's a quote, which I think is fantastic, from Jerry Ledvin, 1976. Jerry Ledvin is the guy who wrote What the Frog's Eye Tells the Frog's Brain. I assume most of you have heard of that great article. Scientific American, I don't remember what year. Anyway, Jerry said, perhaps texture, somewhat redefined, is the primitive stuff out of which form is constructed. And he was actually saying this in an article that's about what he sees in his periphery. So he really was basing this on the same kinds of things that I just documented in these experiments and in this model. Okay, I'm done.
So there's still some time for discussion, and I'd like to start. If your example showed a face, which I'm very interested in face perception, and the face disappeared in the metamer,
yep.
Would the ventral stream response to face that's face specific also disappear? A lot of work on face perception with fMRI looks at responses to peripherally presented faces.
So, so crowding works for faces too. If you put a face in the periphery, if it's big enough and if it's, I kind of want to say simple enough, you can recognize it. But if you put it amongst other faces, you can't. So that basic rule still holds. There's also this kind of, I think the crowding literature tends to be based on letters and objects, and the definition of an object is kind of a funny thing. A complicated object has many features, and so people started in that literature talking about the fact that objects could self-crowd. They could, you don't need multiple objects. You could have a single complicated object, and it crowds itself. So your ability to recognize faces in the periphery does degrade, and it degrades in lawful ways, and I think that this does explain it. The fact that face disappeared is because it's in the midst of a big crowd. If I put that face up in isolation, it would probably still look like a face. If you want, I can show you some examples of that.
Later during
Maybe
the
after?
Coffee break.
Offline. Okay. Andre.
A couple of questions. Right. You didn't mention, or the model doesn't include, as I understand, lateral interactions.
Correct.
You didn't specify like what kind of sample of spatial frequency, orientation, populations you use. So the basic question is this yet another computational model, or does it actually help us to understand how the system works? In other words, obviously that's not how it works, right? So what's missing and how come it works out so well?
Okay, so the model is very, very simplified. Okay, so these are really toy-like descriptions of what neurons are doing. But I think it captures the essentials, the most important piece of the response, and it's enough to produce these metamers. Refining the model, putting in lateral inhibition, putting in surround suppression, putting in things like that, it can't hurt, but you're now talking about a more complicated model. You're talking about more parameters that you have to set or figure out how to select. You're talking about a much more difficult process in terms of generating the synthetic images. So the process of generating the synthetic images requires us effectively to be able to calculate the gradients of the measurements, because we're going to basically do like a gradient descent to push ourselves onto the manifold of images that produce the same responses. So building in more complicated structures into the cell responses will require us to do a lot more difficult calculations. I'm not saying it's not possible. I'm just explaining why we did what we did. So that's the first part of the question. And then you were also asking, I'm kind of separating into one is more complicated features of neural models. And my bottom line there is I think we got the most important piece. I think and the evidence is what I just showed you. It works. We can produce metamers. People can't tell the difference, and we can use that to actually measure or estimate receptive field sizes. I really do think of this as analogous to the color vision story. You make measurements, you notice that people can't tell the difference between these things. You figure out what information they have lost, and that is a prediction, or that should be matched to the underlying physiology. So now we're digging into the physiology to find out exactly whether that's true. So I think, in the sense, I would argue that this is a successful endeavor because it worked, we produced metamers, and it makes predictions that are testable, and we're testing them. We're going in, we're doing the physiology. Oh, also, Jeremy has some MRI on the poster as well. I know a lot of you probably want to see that.
Yeah, so maybe the recognition part.
Okay, right. So how does, when you see something and you say, I can recognize it, what part of your brain are you using? I don't know. I have no idea. Does all the information that you use for recognition have to go through V2? Or have we even captured everything that's happening in V2? Maybe V2 has two sub-areas, one area that does what I just described and another part that doesn't. Or maybe they're not nicely separated in the volume of the brain, but they're actually intermixed, so we won't even be able to see that in the MRI. There are mixtures of cells, some of which are handling recognition and some of which are representing these sort of if you like, statistical averages in the periphery. So I can't answer that at all.
But then the metamer story in color didn't solve the color vision, right? We know that it's more complicated than that.
Absolutely, absolutely.
So your metamer bench test, it's just one among others.
We found a bottleneck, right? We found some information that is being lost by the system. That doesn't mean that we understand how the information that is not lost by the system is being used or represented or being used in later areas. We have not solved that. But the use of this kind of tool, metameric tool, allows you to use external measurements to pinpoint the bottleneck and to really dissect the detailed properties of that bottleneck the same way was done in color vision. Yes, it doesn't explain color appearance, but it had a huge impact, the metamer story in trichromacy, and it allowed all the technology that we have around us, right?
You know, what is the computational goal of the model you described?
But see, I started out by saying I was going to do something a little strange, and let me elaborate on that. I didn't start with a computational goal. And that's not really like me, because a lot of my previous work is about starting with a computational goal and asking how do you solve it. We did the opposite here. We started out agnostic. We said, I don't know what the thing is computing. I'm just going to build in the properties that are there, and then I'm going to ask at the end, what does it compute, right? So it's strange, but that's what we did.
But I was saying, so I want to make a conjecture, and this is that the computational goal is a recognition, and that models like, all the Huber and Wiesel type model, including Fukushima, including our model, would show the same kind of metamers that your model shows.
I think
and
that's...
in fact, there is a poster by Leila that shows that our model, when extended with eccentricity, has, satisfies Buma's law. And
so,
she did not do the metamer thing, but my guess is that probably will make to work. Right?
I think that has to be true.
Back to your texture.
I think it has to be
Jerry
true.
Lattville, right?
It has to be true, because we're talking about the same system, and if you build a system that has simple and complex cells and receptive field scaling, well, those are the main properties that I built in here that gave us these behaviors. There are some details about exactly which measurements you use, and I glossed over these. That is, which linear combinations do you take and which ones do you not take, because there are many possibilities. You have to choose a set, and that will determine in some detail which things are metamers and which things are not metamers, and that's pretty key to this model. So, but the basic structure, the architecture is the same. And I said at the outset, right, this is not, in a sense, everything I told you about that went into this model is not new. They're very old ideas. I mean, Hubel and Wiesel, 62, right? So it's the idea of using some computational glue to put it all together to ask, what are the predictions of the model? What can we do with it? And how can we test it?